Good evening, everyone. My name is Erika Pambianchi, and I'm Dr. Balacchi's graduate student. Today, I would like to introduce you the big picture of what we do in our laboratory. Our lab is interested in studying the effect of pollutants such as ozone, cigarette smoke, and particulate matter on different target organs. The data that you're going to see represent the effect of ozone exposure on the skin. The current skin model that we are using in our lab are three. We have human skin biopsies, 3D models, and 2D models. And the data that we are going to show you represent a study on ACAT cell line, which are human keratinocytes, and keratinocytes make up the 90% of the cells in the epidermis. But why are we interested in studying the effect of the exposure to the different pollutants on skin? It's because, as you know, skin acts like a shield against a huge variety of external stressors, and among them, we know that there are also pollutants. And this is fine, but problems arise whenever in a chronic exposure there is an accumulation of the reactive oxygen species, and so they exceed the physiological level, leading to the development of different skin diseases such as skin aging, psoriasis, and acne. And all of these different skin diseases have as a common denominator an inflammatory status and a redox imbalance. We already showed the link between the development of different skin diseases with the different exposure to pollutants, but now we have a new question. So now we are wondering, what can we do, what can we use to prevent the inflammation and the redox imbalance that is due by the exposure to pollutants? And our answer is trying to use berry extract as a new possible therapeutical approach. We are studying 4 different berry extracts, such as black raspberry, blackberry, blackcurrant, and wild blueberry. And as you know, they are famous, they are known to have antioxidant and anti-inflammatory properties due by their content. So the data that we are going to show you soon, they represent the study of the property of the whole extract of the wild blueberry. But now let me introduce you my student Chad, and he will go through more in details of all our results that we obtained in these 11 weeks.
Thank you, Erika. The goal of our experiment is to study the blueberry properties in preventing the inflammatory and oxidative skin damages induced by pollutants, especially ozone. In the last 20 years, there has been a new structure discovered in the inflammatory pathway called the inflammasome. It's a multi-protein complex, and it serves as a sensor to the environment. After these cells are exposed to oxidative stressors, it then triggers an inflammatory pathway through the inflammasome, which ultimately results in the production of pro-inflammatory cytokines, interleukin-18 and interleukin-1 beta. These cytokines are used as a protective, or using inflammation as a protective mechanism. Here we can test the oxidative stressor that these cells are under. We use a DCF assay, and by the results we see that after the exposure to ozone, there is a significant increase in ROS production. Our blueberry extract can have an effect on these cells, partly because of its polyphenolic composition. Here we have an HPLC provided by the Lila lab, which shows the quantitative and qualitative composition of anthocyanins, where higher peaks represent higher concentrations. To fully understand our blueberry extract, we conducted a variety of different assays and evaluation techniques. To test cytotoxicity, we did an LDH assay. To test the proliferation and migration of our cells, we used a wound healing assay. And to test the inflammation and redox imbalance associated with gene expression and protein expression over inflammasome, we used PCR and Western blot. To test inflammasome-related protein expression and localization, we conducted an immunofluorescence. Our cells were tested under 3 different conditions. We had cells that were exposed to air, cells that are also exposed to ozone, and cells with the treatment of the blueberries. These conditions were set under specific limits. Our experimental cells were pre-treated with the blueberry extract for 24 hours at a concentration of 10 micrograms per milliliter. And our cells that were exposed to ozone were exposed at 0.5 parts per million for 1 hour using an ozone generator. While the cells that were exposed to the air were left uncovered in a chemical hood for that hour. Our first step was to determine an appropriate dosage for our blueberry extract that can be used on the cells that would be non-toxic. Here we have the results for an LDH assay in comparison to Triton, which results in 100% cell damage. We see here that at our highest concentration of 10 micrograms per milliliter, that it was still non-toxic to our cells, so we wanted to go ahead and use this highest concentration because we believe that it would have the highest effect on our cells and the inflammatory pathway. After finding an appropriate dosage, we conducted a wound healing assay between air and ozone, and we see that after the exposure of ozone, it significantly impairs the wound healing process in each time point. And here's a visual representation of that. We see here at time point 36 and exposed to air that the wound is completely closed. But we see after ozone exposure, we see that the wound healing process has been impaired. So we ask ourselves, how can we use the blueberry extract to kind of counteract these ozone exposures? And we can see after the treatment of blueberry that there is a trend where the wound healing process has been enhanced throughout each time point. And here is also a visual representation of that. At time point 36 in air, we also see that the wound is completely closed. After the exposure of ozone, it is still open. But with the treatment of the blueberry, we see that this wound healing process has been enhanced and that the wound is almost closed. Now, to understand the effect that the blueberry extract will have on this inflammatory pathway, we must first understand the inflammasome. After these cells are exposed to oxidative stressors, it then creates ROS in the cells, which then sends signaling to the nucleus to increase gene expression, which ultimately results in the production of proteins outside of the nucleus, which then form the inflammasome structure. These proteins consist of NLRP1, ASC, and procaspase-1. After the oligomerization of these proteins of NLRP1 and ASC, it then recruits procaspase-1, which then cleaves itself into an active form of caspase-1, which then cleaves pro-interleukin-1 beta and pro-interleukin-18 into active pro-inflammatory cytokines. These cytokines are then excreted out of the cells and sent to the area that is suffering from oxidative stress. So, our question is, how can we use blueberry extract to affect the gene expression, the protein expression, and the overall oligomerization of this inflammasome structure? Here we have a real-time PCR result of interleukin-18, and we see that after an ozone exposure at time point 24, there's an increase in gene expression, but with the treatment of the blueberry, we see that this gene expression has been decreased. We also see the same trend of the PCR results of the gene ASC. At time point 24, with the treatment of blueberry, it has also been decreased, and we see the same trend with the caspase-1 gene. Not only do we want to find out the gene expression, but we also tested the protein expression with the Western blot of the protein ASC. At time point 24, we also see that increase in protein expression, but with the blueberry treatment, we see that has also been decreased. We also did a Western blot for NLRP1, and at time point 24, that protein expression has been decreased. Now, we don't only want to study the gene expression and protein expression, but we want to see how these proteins begin to oligomerize and form the inflammasome structure. So we conducted an immunofluorescence where we use dyes to stain certain parts of our cells. We see here that the blue represents the nucleus, the red represents NLRP1, and the green represents the ASC protein. We see here after the exposure to ozone, these colors become much more vibrant, indicating an increase in protein expression. But with the treatment of the blueberry, we see that these colors become faint, which shows that the blueberry is preventing that increase in protein expression. And here the yellow represents the forming of that inflammasome structure. And after the ozone exposure, we see strong indicators of yellow, but not only that, we see the yellow forming outside of the nucleus which is represented by the blue. These strong colors around the nucleus represent the localization of these proteins to form the inflammasome structure. But after the blueberry treatment, we do not see that localization, which suggests that the blueberry treatment is preventing that inflammasome activation. So in conclusion, for the first time, we can see that ozone is impairing the wound healing process and inducing inflammasome activation, and our results also correlate with present literature, which links the increase of inflammatory skin conditions with the increase of air pollution levels. We also see that the blueberry treatment can prevent this inflammatory activation and also induce the wound healing process. So it can be used as a topical and therapeutical approach to counteract the skin damages induced by pollutants. In our future work, we'd also like to test the properties of different berry extracts, such as black currants, blackberries, and black raspberries. We'd also use different exposures of pollutants, such as cigarette smoke and particulate matter. But we not only want to use them in a singular fashion, we want to combine all these pollutants to form a more realistic environment. We also want to use different target organs, such as the GI tract and the lungs, and we would use these different models in 2D models, 3D models, and human biopsies. And I'd like to thank everyone for their support in this P2EP project. And we're at poster number 11 if anyone would have any further questions, or I can take some further questions right now.
Nice work. I'd like to make a plug for purple flesh sweet potatoes also. They make a lot of head signs, but actually, a question. So what do you think the mechanism is that's working to quench this ozone, mechanistically, given what you know about the molecule itself?
So we believe that because of the polyphenolic, so antioxidant compounds that are present in the blueberries, they are able to act against the oxidative species that are produced by the ozone or other pollutants, because they're able to produce ROS. And so, as we have in our cells some antioxidant enzymes, we think that these antioxidant molecules that are present in the blueberry, in this case, they are able to prevent and counteract the reactive oxidative species production due by the pollutants.
I agree. It's curious. I mean, those cyclical structures, they'll absorb all of that reactive oxygen pretty easily. So, yeah, cool. Thank you. Nice work.
Thank you very much. And I'm sorry because I've seen that some pictures were not in line. I don't know what exactly happened when we upload the presentation. I'm sorry for that.